So good morning. Thank you for joining us today for the inaugural Penn State 3-Minute Thesis competition hosted by the Graduate School. I am LaVon Esters, Vice Provost of Graduate Education and Dean of the Graduate School. I'm also a proud Penn State graduate, so we all have a shared identity here, being Penn Staters. I received a PhD in Agriculture and Extension Education in 2003, and I'm thrilled to be back as the executive champion for Penn State's incredible graduate students and educational programs. In the spirit of today's event, our planning committee has asked me to keep my remarks to 3 minutes, so I will be brief. Luckily, they told me that I'm not being timed by our presenters. First, I would like to thank our partners for the event, the Graduate and Professional Student Association and the Graduate School Alumni Society. I would also like to give special thanks to our volunteer judges, Dr. Kathy Drager, Associate Dean for Research and Graduate Education in the College of Health and Human Development and Professor of Communications, Sciences, and Disorders and former Interim Dean of the Graduate School. Also, we have Dr. Bimal Balakrishnan, Penn State graduate and professor and Associate Dean for Research in the College of Arts and Architecture at Mississippi State University. Bimal is also President-elect of the Graduate School Alumni Society. And we have Dr. Esther Abonyo, Associate Professor of Architectural Engineering and Director of Penn State's Global Building Network, and Priyanka Paul, doctoral student in Human Development and Family Studies. The graduate school's primary mission is to support the holistic success of all graduate students at Penn State. Events like these are an important professional development opportunity that helps students refine their communication and presentation skills and practice highlighting the impact of their research and scholarship to diverse audiences. A vibrant education enterprise is crucial to the status of Penn State as a Research One institution, and graduate students like yourselves and those in the audience help shape the research output of an institution. They engage in innovative projects, collaborate with faculty, play a vital role in groundbreaking research, and mentor undergraduate students as well. A robust graduate education program attracts top-tier faculty, like those I just shared a little bit ago, and students, and fosters a culture of intellectual curiosity and scholarly achievement. This, in turn, positions Penn State as a hub for groundbreaking research and a leader in higher education. There truly is a reciprocal relationship between research and graduate education. Simply put, graduate education and students play a critical role in Penn State's success. So, thank you all for being here to support our graduate students by being actively engaged in the community. We are all in for a treat through these dynamic, exciting, and brief research presentations. And I will say before I go to my last paragraph that this has been something we've been thinking about, and I cannot tell you how excited I am to be here today for this inaugural event. So I'm really proud of all 11 of you and those who are supporting you all today. So now I'm going to hand things over to Leslie Jackson, the Graduate School's Associate Director for Graduate Student Career and Professional Development, who will be our MC for today's competition. Leslie.
Good morning. Thank you, Dean Esters, for starting us off. I, too, will be keeping my comments to 3 minutes today. I'd also like to thank our judges for taking the time to be here today and providing our students with excellent feedback. I'd like to again acknowledge our partners for this event, the Graduate School Alumni Society and the Graduate and Professional Student Association. Lastly, I want to acknowledge the 3-Minute Thesis Committee. I'm privileged to work with a thoughtful, talented, and incredibly hardworking team of colleagues. And it doesn't hurt that this group has a great sense of humor. Thank you, Heidi, Jeff, Jen, Jillian, Liam, and Lisa. The story of the 3-Minute Thesis starts in a drought-ridden Queensland, Australia, where the residents were desperately trying to conserve water. Many Australians had 3-minute egg timers attached to their shower walls for this purpose. Many of us have our best ideas in the shower. And during such a moment, the then dean of the graduate school Emeritus Professor Alan Lawson had the idea for the 3-minute thesis, and the rest is history. Today, 3-minute thesis competitions are held in over 900 universities across more than 85 countries worldwide. And while an exciting aspect of these events is often the awards, participating in 3-minute thesis develops essential communication skills and a candidate's ability to effectively explain complex ideas in language appropriate for a general audience. If you're unsure why this is an important skill, you only have to look to a small healthcare issue we had in 2020 and how important it was for healthcare professionals to communicate effectively with the public. For our competition today, our students will be competing for 3 awards. First place for $1,000, second place for $500, and our People's Choice Award, sponsored by GPSA, for $500. As audience members, you'll vote in our People's Choice Award category via a QR code. This link will only be open for 10 minutes at the conclusion of the presentations. Our participant who takes first place will represent Penn State at the regional 3-minute thesis competition for the Northeast Association for Graduate Schools. Our 11 presenters represent 5 different academic colleges and 9 programs of study. I guarantee you will learn something from these presentations today. Having spent some time with these students this past week, I know that beyond their impressive academic work, these are some incredibly special individuals. They are caring and warm. They are dedicated to and passionate about their craft. They are humble and resilient and truly looking for ways to make a positive impact on the world around them. In short, they are Penn State graduate students. I'm sure you are as excited as I am to welcome our wonderful students to the stage today. Thank you. Before I welcome our first presenter, if you have not already done so, please silence your cell phones. And we ask you to remain seated during the presentations. If possible, wait for the 2-minute judging period between student presentations before moving around the room, leaving or entering the facility, so as not to disturb our presenters. So, without further ado, please help me to welcome our first presenter. And I just learned this morning there's a bit of a change here. Dr. Yue Yen, who is not a doctoral candidate anymore in biomedical engineering.
Cancer, it's something we're all too familiar with. Bladder cancer in particular stands as the 6th most prevalent cancer in the United States, with over 80,000 new cases each year. 75% of them are non-muscle invasive, meaning the tumor has not penetrated that far into the muscle layer. The primary treatment for these patients includes a surgical procedure known as transurethral resection, followed by BCG treatment using a live attenuated strain of tuberculosis. You heard me right. It's been the gold standard for the past 34 years, ever since the 1990s. The challenge is, however, 40% of patients do not respond to BCG. Some of these non-responders, unfortunately, cannot survive, and for many others, the only remaining option is complete bladder removal. And these people are exactly why researchers like myself are actively diving into new solutions. So really inspired by recent advances in immunotherapy, my work explores the use of microbial products, which are beneficial substances derived from microorganisms, but without any live bacteria. You can think of them as helpful byproduct of these tiny organisms that can fight cancer. And broadly speaking, over the past few years, we've identified several microbial product cocktails, MPCs, for bladder cancer treatment. What's most exciting is that we're able to double the long-term survival rate in mice treated with our MPC compared to the standard BCG. So here's how it works. Imagine a patient arriving in a clinic diagnosed with non-muscle invasive bladder cancer. Instead of facing the quality of life impact from chemotherapy or the risk of ineffective BCG treatment, living the rest of their life without a bladder, there's now an alternative, MPC intravesical therapy. This directly delivery of liquid MPC into the bladder through a catheter. And of course, no single drug agent works for everyone. Therefore, we have developed a eye care assay. This microfluidic platform is designed to predict the efficacy of MPC or BCG treatment in less than 24 hours in a laboratory using a tiny biopsy sample taken during surgery. The most effective MPC from a panel of 32 previously determined by our AI-assisted workflow to boost the immune system to fight cancer will be identified. This aids doctors in making informed decisions, improving patients' chance of survival. More research is needed to bring this to clinic. Our work not only pioneers a novel approach to bladder cancer treatment, but also sets the stage for a new era in personalized medicine. Beyond bladder cancer, this method has the potential to revolutionize the rise of cancer. Our vision is a world where cancer treatment is not one size fits all, but tailored, improving survival and quality of life for patients globally. Thank you.
Please welcome our next presenter, McKenna Lenover, doctoral candidate in anthropology.
My stomach hurts. That's something you may say a lot in your day-to-day life, or maybe even here. Digestive issues have become increasingly commonplace in our society. But what if I told you there was a more serious explanation at play? Irritable bowel syndrome, or IBS. IBS is a chronic digestive condition that involves symptoms of abdominal pain as well as changes in stool habits like constipation or diarrhea. It affects 20% of the United States. That means potentially 2 in 10 people in this room are suffering from the condition right now. Speaking in front of others today may be upsetting your stomach. It's certainly upsetting mine. That's IBS in a mild form. But what if the pain was so debilitating you couldn't even show up? That's the reality for many with IBS, which shows why it's essential to get to the crux of this condition. Here's where anthropology can come in. IBS falls within a framework called diseases of industrialization. Those are diseases whose prevalence increases alongside changes in behavior that we see as we adopt industrialized lifestyles. Let me set the scene for you. You're at work. You're probably sitting all day. You might not be getting much physical activity, and you're probably not getting outside exposed to vitamin D. The stress is probably pretty intense at times, and it may even keep you up at night. During the busyness of work, you might be eating a quick processed lunch. This is the type of behavior we're talking about. Some other diseases of civilization include diabetes or heart disease. My work takes an evolutionary medicine approach to study IBS, trying to understand how our rapidly changing environments around us mismatch with our slow-evolving human biology. To study this, I'm sending a survey out to Philadelphia area residents, asking about their digestive health and their behaviors. I'm then statistically analyzing these responses to determine how their differences in behavior contribute to their differential disease status. So far, we see about a 32% prevalence rate of IBS in the Philadelphia area, and some of the most significant predictors of having IBS include eating low-fiber diets and experiencing a lot of anxiety or stress. One of the most exciting parts of this research is how we can use anthropology to better people's health and wellness. Right now, there's no one treatment or preventative measure for IBS, and clinicians are desperately seeking ways to help patients. It's clear behavior is driving this illness, and my study is one of the first to determine just in what direction. We can use this to inform interventions so we can then lower patient IBS prevalence, prevent future IBS, and we can do it all with the power of anthropology. Thank you.
Please welcome our next presenter, Paul DiStefano, doctoral student in cognitive psychology.
Is a hot dog a sandwich? This simple question has ignited a passionate debate across the internet. People fervently argue whether a frankfurter on an open-faced roll should be classified as a sandwich. But as a psychologist interested in creativity and knowledge representation, I am interested in what factors influence how someone categorizes a hot dog. To me, this debate demonstrates the intricacies of human language and individual differences in how people organize information mentally. What if the hot dog sandwich debate reveals something profound about the organization of information in the human mind? What if our creativity influences how we categorize things, which in turn affects how we interact with the world? These questions were among my influence for my research on overinclusive thinking. Overinclusive thinking is the cognitive process where individuals tend to include irrelevant or extraneous information. In my work, I quantify overinclusive thinking by measuring how individuals categorize everyday objects. I developed and validated a new psychometric tool, the Categorical Overinclusive Thinking Task, or COVERT for short. In this task, participants make 204 categorical decisions across 9 categories. For instance, is mathematics a science? Is a photograph a tool? These seemingly trivial decisions may hold clues about the underlying structure of semantic memory. Your semantic memory is like a library of different concepts, and like a library, your semantic memory is organized into different genres or categories. An individual who is more overinclusive may be able to think of different genres more easily because the shelves of their mental library are closer together. As a result, we may view this person as more creative. Across 3 studies, sampling a total of 750 Penn State undergraduates, my research found that covert scores correlate with traits associated with creative thinking, such as personality trait openness, creative behaviors, and creative achievements. This novel measure stands as the first rigorously tested measure of overinclusive thinking, offering a reliable tool for researchers and clinicians alike. And as a brief aside, we also found that 50.1% of our sample classifies a hot dog as a sandwich. So the debate lives on. Fortunately for me, these debates fuel my research interests. In my future research, I will investigate how covert scores across different creativity levels influence the structure of semantic memory. This way, we can understand the individual differences in how people mentally structure concepts. This work will deepen our understanding of knowledge representation and its influence on creative thinking. Thank you.
Please welcome our next presenter, Auja Bywater, doctoral student in food science and technology.
As farmland continues to decrease, how do we produce enough food to safely feed everybody? Hydroponic farming is a growing method that allows us to grow food in non-traditional farmland settings. Instead of needing acres of land, food can be grown indoors, in vertical configurations, in cities, and year-round. Instead of needing soil, food can be grown in water, enhanced with nutrients for the plants, often referred to as nutrient solution. Instead of needing mass amounts of water, these systems often recirculate and thus conserve water. This is an exciting way to provide everybody with fresh, healthy food. However, just like traditional farming, there are food safety risks. Bacteria have been found on produce grown in these systems that can make people really sick. Since it's a relatively new field, especially compared to traditional farming, there is limited research and understanding bacteria in these systems. To add to that, there are different types of hydroponic systems that all influence how the plants, roots, nutrient solution, and bacteria interact. This presents complex systems that need to be studied to understand these relationships and keep our food supply safe. I wanted to look at how these different types of hydroponic systems influence the bacteria present. So I worked with plant scientists at Penn State to grow bok choy in the 5 different hydroponic systems that you can see here on the screen. The Kratky and deep water culture system had the plants and their roots rest in a nutrient solution. The drip irrigation nutrient film technique, and ebb and flow systems actively apply nutrient solution to the roots in different ways that you can also see here on the screen. I sampled the nutrient solution as well as a harvested bok choy to see what we would find. I found that the bacteria present was influenced by the type of hydroponic system. The drip irrigation and ebb and flow systems used a soilless media to physically support the plant's growth. Compared to the other three, these two systems had higher counts of a bacteria that we associate with the environment. This suggests that the materials and methods used in these systems can influence the bacteria present. When we tested the harvested bok choy, we did not see a difference in bacterial counts. But I want to point out that this was a small-scale study with very controlled practices that may differ from commercial production. It's so important to stress that although the bacterial differences in this study didn't transfer to the plants, they do have the potential to, especially in commercial production. This research shows that the different types of hydroponic systems do influence the bacteria present. Understanding that these systems have different risks will allow for tailored food safety practices needed to keep us all safe.
Please welcome our next presenter, Aditya, doctoral candidate in biology.
Communicable diseases, or diseases that can spread from person to person, have a huge impact on our society. Even before COVID-19, which has already killed about 7 million people so far, communicable diseases accounted for a quarter of the total burden of human disease on Earth. Some diseases, such as mumps and measles, have safe and available vaccines, and we've made a lot of progress vaccinating more and more people in recent years. Take measles, for example. We went from 15% of kids being vaccinated against measles to 80% worldwide in a span of 4 decades. And that's amazing. And that should mean that we have effectively eliminated or stopped the local transmission of most infectious diseases at this moment in time, right? Well, unfortunately, that's not the case. Let's go back to measles for a second. We have 80% of kids worldwide vaccinated, and a vaccine that's basically 100% effective. And yet, we saw 10 million cases and 100,000 deaths just last year. So why is it, with such effective and safe vaccines, with such high vaccine coverage, we still see so much disease? Well, and what do we really want to know next in order to actually eliminate these diseases? Well, first we want to know, where are the unvaccinated individuals? Current measures of vaccination coverage are either really out of date but accurate, or really inaccurate and available in a timely manner. I worked with the World Health Organization to look at aggregated healthcare data to develop a method to predict vaccination coverage at the subnational level that's both timely and almost as accurate as the gold standard that's often out of date. But disease can persist even when you have a lot of vaccination, whether that's due to pockets of local transmission or a lot of introduction of cases coming from elsewhere. And I developed a methodology to actually study what was happening. Was it more local or was it more regional and widespread, using historical data from the United States? And what this allowed us to do is to target vaccination campaigns and target interventions that are better suited for whatever scenario is happening in a given place. And third, we need to know how much progress has already been made towards disease elimination. Here, we looked at different data from Nigerian states, and we saw patterns of incidents, i.e. the number of cases, that differed from year to year. And we were able to characterize these patterns and get a good idea of how far a place was to actually eliminating the disease, and importantly, how much more effort was still left to go. Basically, in terms of the diseases that we are looking at, in terms of communicable diseases, I hope that the methods that I've developed can really help with supporting communities in eliminating these communicable diseases once and for all, even in places where data are lacking or data are of low quality. Thank you.
Please welcome our next presenter, Shabnan Rahimnejad, doctoral candidate in mechanical engineering.
couple of years ago, if someone came to me and gave me a news that a professional athlete got Achilles tendon rupture, my answer was rather simplistic. Surgery, followed by casting or use of wood, then physical therapy, and then we will have a full recovery. However, working with patients and athletes who are doing their best to recovery changed my mind. Because some of them are doing their best, but at the end of the day, they're just struggling without any improvements. Even there are some who are forced to retire from their professional careers due to this problematic injury. And as a former professional athlete, I understand the difficulty of saying goodbye to sport. Analyzing the data of the images brings me more ideas. The images shown here are related to the patients who got ruptured at least 3 years ago. And as you can see, there is fat filtration and muscle shrinkage and fibrosis when we compare the injured leg to the healthy one. And some of these might be due to the lack of use of muscle during the weeks after surgery. And this brings us a research question. Can we modify the rehabilitation method to fight with these problems? Before talking about the aim of my study, let's talk about this small device. I believe some of you have seen electrical stimulation device, a device with the pads that are attached on a skin and stimulate the muscle and apply load on the muscle. We hope by use of electrical stimulation muscle, in addition to the usual conventional physical therapy, we can fight with the shrinkage of the muscle. Our results are optimistic. So we have 17% muscle shrinkage in our treatment group compared to the 23% in control groups. And we are still gathering the data. In conclusion, we hope the result of the study help the patients who are suffering from Achilles tendon a smoother recovery. Then there would be no athletes who say goodbye to their professional careers due to this problematic injury. Thank you.
Please welcome our next presenter, Aditya Sapre, doctoral candidate in chemical engineering.
Do you guys know that out of 10,000 candidates, only 2 to 3 drugs get the final approval of launch after a long and strenuous process? The major challenge in drug discovery is identifying the right type of protein that can bind with the right drug. Currently in industry, these two processes are done separately. But before I tell you how we combine these two in one single continuous high throughput process in our lab, let me reintroduce myself. I am Aditya Sapre. I am a 4th year chemical engineering graduate student. Current technique involved around protein identification make use of physical properties of proteins such as its shape, size, and density. This makes it difficult to harvest the right type of protein from a complex mixture of proteins having very similar physical properties and in quantities that are sufficient for the tail end analysis. Now here, my channels come into picture which are shown in the slide. These channels have length equal to my forefinger, and subchannels present there have width and height equivalent to 1/100th of a single grain of rice. So you can imagine how tiny these channels are. In these channels, we pass a complex mixture of proteins, and each type of protein is colored with a different dye as well as different candidate drugs. As I, or most of us, move towards the delicious smell of freshly baked cake, one of the protein from this mixture moves towards the correct candidate drug and binds with it. Because of these pure binding interactions, we were able to separate the right protein from this mixture very easily and effectively. The type of characterization technique that we use to analyze the output of this process is called UV visible spectroscopy technique. It's a technical jargon, but let me simplify it for you. It's a very simple technique that shines light on these proteins. And as I mentioned before, each type of protein is colored with a different dye. So each color has its own unique signature, which allows us to identify this separation very simply and effectively. Now, the type of proteins that we are studying lie under a family called kinases. And why we are doing that? Because these proteins are crucial for cell growth and proliferation. Some advantages of my process: it requires minimum protein quantity, it requires minimum supervision, and it just gets done in 30 minutes, while all the industrial processes take about 5 to 6 hours. So at the end of the story, if we scale up this process to an industrial level, it will speed up the early stages of drug discovery and make it much more cost effective. I am Aditya Sapre, and that is my pitch. Thank you.
Please welcome our next presenter to the stage, Tahia Taranam, doctoral candidate in Civil and Environmental Engineering.
Have you ever come across news on fish dying due to harmful algae growth? This happens when there is excess nutrients like nitrogen and phosphorus in water. A major source of nutrient pollution is runoff from agricultural lands. Synthetic fertilizers are fast dissolving and often ends up into the runoff. The problem with using dairy manure as a fertilizer is its high nutrient variability, meaning chances are we are often over-applying it. Apart from degrading the water quality, use of fertilizer also increases nitrous oxide emission from soil, which is a much more powerful greenhouse gas than carbon dioxide. But we still need to use fertilizers to meet the increasing food demand. So what if we find a fertilizer that can maintain crop production while minimizing environmental consequences? To do so, we use the key concept of ecological engineering in our lab, which is finding a sustainable solution in nature. And one such solution is duckweed, the image in the slide. It's a tiny aquatic plant. You might have seen them floating in ponds, aquariums. What's really cool about duckweed is it can grow in wastewater and uptake nutrients. And it has the perfect carbon and nitrogen ratio, so we can use it as an organic fertilizer. And using duckweed, we can upcycle nutrients back into agricultural lands. I have been testing duckweed with other conventional fertilizers in controlled laboratory environment. I built these soil columns in the greenhouse, like the diagram on the right, applied duckweed in some, a synthetic fertilizer and dairy manure in some others, and I grew a crop. Now I had labeled the nitrogen in the duckweed and the other fertilizer to trace its movement in the system. Think of this like adding a dye in a water pipe to trace a leak downstream. This technique helped me to pinpoint what proportion of the fertilizer was lost as pollutant, be that some nitrous oxide emitting from soil or nutrient leaching down the soil column with water compared to plant's uptake and what remained in soil. So as the plant grew, for 3 months every week, I simulated rainfall on them and took gas and water sample to measure those losses. So far, I have found that the duckweed fertilized soil lost less nutrient than others, and it supported crop yield similar to the synthetic fertilizer. This is because the duckweed degraded slower. My research can help make a case for farmers to switch from using conventional fertilizers to using duckweed. And if duckweed is widely accepted, this simple nature-based solution can reduce the environmental footprint of using fertilizers. Thank you.
Please welcome our next presenter, Abby Gantz, doctoral candidate in anthropology.
For as long as humans have been walking on this planet, diseases have been hounding our footsteps. They have prospered in our religions and flourished in our narratives, tantalized scientists and mystified historians. They have impacted every single human being in every single population, including probably many of you here today. And they have not done so equally. Research shows us that individuals from destitute, disenfranchised, and discriminated against backgrounds suffer and perish from diseases at disproportionately high rates. And despite decades and even centuries of research into this topic, we're still only scratching the surface of the various determinants of health inequalities that underlie these epidemiological trends. We've been studying some determinants of health for the last couple of decades, for example, sedentism, unhealthy diets, inability to access healthcare, but other determinants of health inequalities are only now beginning to be explored. My research is on one of these understudied yet critical determinants of human health, specifically the human oral microbiome. The oral microbiome is the collection of millions of tiny organisms that live within our mouths. And these are not random interlopers. They have been co-evolving with our species for millions of years. And because of these tiny organisms play vital roles in our biologies, including detoxifying environmental chemicals, protecting us from infectious diseases, and even increasing the amount of nutrition we can derive from our foods. Now, while these microbes are usually very beneficial, when things go wrong and these microbes get out of balance, things can go really wrong. And this same oral microbiome has been strongly associated with diseases including diabetes, dementias, and cardiovascular heart disease, which are some of the biggest killers in the United States today. That is why, with my doctoral research, I focus on the question of how has the oral microbiome's relationship with health and disease evolved over time, and what implications does this have for health disparities for us? To study this, I look at calcified dental calculus. What this is the same biofilm that the dentist removes from your teeth, but I look at it not in modern people, but in archaeologically excavated human skeletons. I take this substance and using ancient DNA methodologies, I carefully reconstruct the genetic code of the microbes that have been trapped within for hundreds of years. Now, with this research, I have managed to describe previously unobserved distinct microbial ecologies in ancient populations and really measure how diets, lifestyles, and behaviors have modulated the disease outcomes in the past in ways that have resulted in health inequalities. Now, this research isn't only useful for understanding the past, but is also vital for today. Our team is putting together a new type of medical therapy called oral microbiome transplants, which could greatly alleviate the oral and systemic disease risk of vulnerable populations today. Thank you for listening.
Our next presenter is Jorge Diaz Gutierrez, doctoral student in civil and environmental engineering.
So, who loves being stuck in traffic? Okay, honestly, I can't be the only one who loved the adrenaline of rushing late to the airport and see a huge line of vehicles right in front of you. I mean, wondering whether you're gonna miss your flight because you still have to check your bag, go through TSA, or rush the whole airport, and being on time is amazing. However, despite all the fun that you can have, congestion has a lot of downsides for the airports, people, and the environment, and my master's thesis had to look for a solution. The idea we came up with is something really simple. If you have one side of the airport that is completely congested, let's say departures, while arrivals is empty, why don't you turn on a sign and tell people, hey folks, it's the same terminal. Instead of going to departures, go to arrivals and just use the stairs. In this way, people will save time and you will manage your congestion. So this idea is really simple, easy to implement, and low cost for the airport. And the real question was when to turn on and off the sign. So we tested 16 different strategies, and we used simulation with 80 different traffic conditions at the airport in Seattle. Then we compared the results against a baseline where we didn't have any traffic sign activated. In this way, we know how much time you were spending in that congestion, how many people is trapped in that congestion, the amount of CO2 emissions, and how many people was getting in and out of the airport. The comparison we used was with repeated measures ANOVA, so these results can be used in any airport in the States. We found that the tool is great. Honestly, most scenarios were perfect. In particular, when you activate the sign before the congestion starts in departures, meaning that you delay the congestion, and you deactivate before the congestion starts in arrivals, meaning that you avoid the congestion in arrivals, you have the greatest benefits. However, it's not always a good idea to use the tool, because if both sides of the airport are congested, everything is going to get worse. Now, with the results of a master thesis, I'm doing an internship with a national lab. We are developing an algorithm that makes these decisions when to activate and when to deactivate automatically. So any airport can display the sign, run the algorithm, decrease the congestion, and you don't have to worry if you're going to miss your flight. Thank you.
Please welcome, last but not least, our next presenter, Kalei Kowalczyk, doctoral candidate in nursing.
Death can be emotional, scary, and beautiful. Death is often an uncomfortable topic for many due to the stigma associated around death and dying in the United States. However, this barrier ends up creating issues, causing people to avoid talking or even thinking about their end-of-life care choices. But at the end of the day, everybody dies. As a hospice nurse of nearly 4 years, I'm often told that it takes a special person to do the type of work that I do. But does it really? Recently, the AACN recognized that undergraduate nursing students need to be proficient in both hospice and palliative care upon graduating. However, students still have limited opportunities to access educational and clinical resources around end-of-life care. To be able to create a better paradigm of care for ourselves and our loved ones, we need to be able to talk about how end-of-life care makes us feel as people and as professionals, to be able to integrate it better into nursing education. For my dissertation, I used qualitative descriptive design study to look at 21 undergraduate nursing students' experiences providing end-of-life care and how these experiences influence how they feel and their emotions. This picture here behind me shows undergraduate nursing students' emotions that they felt when they either thought about or provided end-of-life care to their patients, families, or even their loved ones. Students felt many positive, negative, and even visceral emotions such as anger, anxiety, sadness, discomfort, relief, peace, and even grief. As students progress through their nursing programs, they begin to harbor these feelings as they experience death. As they harbor these feelings, it can cause students to have or experience distress or have unmet emotional needs. Many of these emotions, as you can see, are influenced by many different factors, such as their academic experiences, clinical, professional, or work experiences, or even a personal encounter they might have had with death in their family. Some of these experiences we could influence as educators. To begin breaking the stigma around death and dying in the United States that's provided to ourselves and our loved ones, it's important that we acknowledge the following. It's important that we acknowledge that undergraduate nursing students have emotions, especially after caring for dying patients. It's also important that we understand that there are many experiences that influence how undergraduate nursing students feel. And lastly, as educators, it's important that we normalize that students have these emotions, but also develop resources to help support them and process these feelings. So I leave you with this question. Does it truly take a special person to provide end-of-life care? Or do we have to better educate our students and prepare them to experience death at the bedside? What are your thoughts? Thanks.
So yesterday we had the research exhibition, and I got a chance to walk up and down every aisle, talk with students, and then sitting here today observing our inaugural cohort, if you will, of students who participate in this 3MT. You know, I'm a Penn State graduate. I earned my PhD from Penn State, and I'm excited to be back here. I mean, I love Penn State. It did a lot for me, but having attended the exhibition yesterday and being able to listen to you all today, we have some talented freaking students. Let me just say, so let's give them a hand, please. I had to emphasize like that because that's really how I feel. I just, just being able to listen to every presentation today. I mean, this is phenomenal. I mean, being able to know that, we had this vision to create this, and now to see it come to life, and to know that you participated in it, I mean, I can't ask for anything else. And so it just really makes me proud to be the dean of the graduate school at Penn State, and to know that I come to work every day, and I'm in the midst of talented folks like yourself, and even those grad students in the audience today. So I just felt it necessary to say that, because that's how I truly feel. I mean, we have some talented students, and you all are going to do great things. A lot of what we saw today, I can't wait for it to, to change lives, right? And so that's what it's all about, making an impact. And so there's no doubt in my mind, each of you would do that. Each of those who I saw yesterday would do the same thing. So I just want to take a moment and share that with you, because that's how I feel about you all today, okay? Thank you. Okay, so this is the moment we've all been waiting for, right? Waiting with bated breath, as they say, for the announcements. So what I'm going to do is I'm going to announce the winners. I'm going to start with People's Choice, then I'll proceed to second place, I believe. Is that right? Yep, second place, and then I'll go, my last one will be the first place award. So people have selected Paul DiStefano, if I said it correctly, as our People's Choice winner. So, congratulations. You want to stand up? Thank you. Thank you so much. All right, our second place winner is Auja Bywater, doc student in food science and technology. Congratulations. Here you go. Thank you. We don't have any drums, Leslie. Right? Thank you. I appreciate that. Good. All right, you got that. So our first place winner is Aditya, Sapre, doc candidate in chemical engineering. Congratulations. Congratulations. So I'm going to turn it back over to Leslie, who's going to close us out. Thank you.
Well, this is our first 3-minute thesis event, and I think what made it so successful were the wonderful students that we had here today. So I'd like to give all of them another round of applause. And of course, all of you that supported them, whether you were here in person or online, I thank you so much for coming and supporting them and being here in whatever form. When I said earlier that this was a special group of students, I truly meant it. Every single one of them, beyond the research, beyond what they're doing academically. They are really a wonderful group of students. All of our graduate students are really special in a lot of ways. I'm so happy to be able to work with these students. So I hope you attend next year. We plan on doing this every year. And support us again. But thank you all for attending and coming. Again, thank you to our judges and our GPSA helpers. Everybody have a wonderful rest of your weekend. Thanks so much.